My name is Kim Raineri. I'm the Chief Technology Officer for Aspen Neuroscience. I'd like to thank the organizers for allowing me to speak today and sharing a little bit about Aspen Neuroscience and the work that we've been doing. Let me see. All right. So Aspen Neuroscience is a Series B company and a spin-off from Scripps. One of our founders, Gene Loring, is actually here today. Hi, Gene. We're well capitalized and from a group of top-tier investors, and we raised almost $150 million last year. Our focus is on iPSCs, autologous iPSCs, induced pluripotent stem cells, as a platform. And we've developed proprietary AI machine learning bioinformatic methods to characterize the cells that we create and we produce, which enables us really to pick the best cells for the patient. We have our own in-house manufacturing, as well as quality units so that we can control our own destiny and really hold our IP close. Our lead program, ANPD-001, is in Parkinson's disease, and our vision is to be the leading autologous iPSC cell therapy company. So the big news that we have is that we recently cleared our IND, enabling our first-in-patient studies for an autologous induced pluripotent stem cell-derived dopaminergic precursor cell for the treatment of Parkinson's disease. And so now that we've transitioned to a clinical stage company, we're executing on that platform. Our interactions with the FDA have given us some really good insights onto approvable endpoints for future pivotal trials. We have a patient screening study that is ongoing with multiple clinical sites. And in that patient screening study, we're using DHT, or Digital Health Technologies, in partnership with Rune Labs and Emerald Innovations. And we're hoping and planning to dose our first patients next year. On the manufacturing side, we have received manufacturing clearance from the California Food and Drug Branch, and patient material manufacturing is already underway with several patients complete. And we've expanded our manufacturing even further beyond from our pilot plant and have qualified additional space to increase our clinical capacity. And we're also executing on a strategy to industrialize our manufacturing platform, and I'll tell you a little bit more about that later. And so now I'll tell you a little bit more about our platform and some key elements. So Aspen has built a platform that can harness the power of autologous cell therapy to potentially enable long-term engraftment and efficacy without immune suppression. With advances that we've made in iPSC technologies, we can now make high-quality pluripotent stem cells from any person for the generation of autologous cell sources for any cell type in the body. We've amassed a deep expertise in the generation of consistent, high-quality iPSCs and the creation of genomic tools to ensure safety and quality of cells at every stage of our manufacturing process. And the use of genomics assays like Pluritest and NuriTest are what set Aspen apart from others in the field and will really enable our autologous approach. And lastly, we've developed a proprietary delivery device that allows for very precise, ultra-low volume delivery of cell products into the CNS, which can be used under MRI guidance. So what you typically hear is that variability between patients is perceived as an obstacle for autologous cell therapies. And in contrast, autologous CAR T therapy, where you see large patient-to-patient variability due to the starting material, the process of reprogramming the cells, the adult cells, provides the opportunity to start with a consistent product from each patient. And the reprogramming process rejuvenates the cellular machinery by elongating the telomeres and restoring lysosomal and mitochondrial function. The reprogramming process that we use produces many more clones than are needed for creating the cell therapy. And a key part of our process is to select the appropriate clone that will minimize variability in the manufacture of the differentiated product. We found that the best indicator of iPSC clone quality is to take an unbiased machine learning approach to analyzing the whole transcriptome data, which is accomplished by pluritest. The pluritest is a common assay used in the field to assess novel pluripotent lines and identify those that are capable of forming any cell type. We've taken the same approach to create a whole transcriptome profile for the dopaminergic neuronal precursors, which have demonstrated efficacy in preclinical models of Parkinson's disease. This is an important step because the cells must be transplanted as precursors and require maturation in vivo to become functional dopamine-producing neurons. NeuriTest really provides the confidence beyond the typical QC panel, like flow cytometry, that a novel batch of precursors will behave as expected after transplantation. And in the development of our autologous cell therapy products, we generate vast amounts of data from numerous patient-derived cell lines, including many clones per donor at different stages of our manufacturing process. And we can take advantage of this data generated by the many cell lines to understand subtle differences in the cell's genomic characteristics and to create predictive models for downstream cellular behavior. And we're using this data to evaluate key efficacy characteristics like engraftment, innervation potential, dopamine production, and long-term durability. And this is helping to inform our critical process parameters and our critical quality attributes for our intermediates and our drug products. And what we're working on now is how to automate and industrialize the manufacturing platform. And so as I mentioned earlier, we've manufactured for our first few patients at our pilot facility in San Diego and have recently completed the build-out and qualification for our new facility in Torrey Pines, just up the street. This facility provides more clinical capacity and has additional room for expansion, which we're planning to use for our process automation. We've completed a broad search and evaluation of technologies, solution providers, and development partners, and selected some key systems and partners that we're going to take forward into our process industrialization program. I hope to be able to share more on that in the coming months, but what we're intending is that this program will be ready to launch for our Phase 2B program. So now I'll talk a little bit about Parkinson's disease. And, our long-term plan is a stepwise approach. Sorry about that, I missed a slide. Our long-term plan is a stepwise approach to move from the aseptic manual R&D process to a semi-closed, semi-automated process, and finally to a process that is end-to-end closed and automated. And the challenge that we have is not to produce large quantities of cells for each patient, but to produce small quantities of cells for many patients in parallel. And we're developing this template for automation that gets us into the multiple hundreds of patients that we can then use as a template for the scale out to the thousands of patients. Now I'll tell you a little bit about Parkinson's. So Parkinson's disease is the second most common neurodegenerative disease in the world, and it affects millions worldwide. It's really the loss of a single cell type and a localized part of the brain that is responsible for the development of the disease symptoms. And by the time of diagnosis, it's estimated that the majority of dopamine-producing neurons in the substantia nigra have already been lost. and there's no intervention that can prevent further degradation of those dopamine-producing neurons. But there is a history of cell replacement therapy that informs our approach to replacing dopamine-producing neurons. And that's fetal dopamine neurons that have been previously used as a source for cell replacement for Parkinson's disease. And these are the proof of concept studies that have provided valuable information on which type of patients are appropriate, how many cells must survive for benefit, as well as how and where to deliver those cells. And though there's many disadvantages to the fetal cells and sources, which make that type of therapy impractical, some patients did actually have significant graft-related benefits for years after a single dose of fetal cells. But this was only accomplished with immunosuppression. And with an autologous approach, we believe immunosuppression is not needed. And this is what we believe one of the key advantages of autologous iPSC therapy. Immunosuppression can be very problematic for older patients and patients with comorbidities, which is really the typical profile for a Parkinson's patient. This approach has been further reinforced by a study that was published in 2021, which was a head-to-head comparison performed on NHPs by Marina Emberg at the University of Wisconsin. In a 2-year study without immune suppression, autologous cells from the monkey's own iPSCs were able to integrate within the host brain and resulted in benefits in the clinical rating score, which persisted for the length of the 2-year study. And for the first time, there were reported benefits in non-motor behavior only in the autologous group. And this study reinforced the need for immune suppression for allogeneic cell therapy, even as the cells in the brain did not provide clinical benefit and did not provide robust fiber outgrowth without immune suppression. Which leads us to our IND-enabling work and our IND-enabling study, where we demonstrated on the safety side that the cells do not migrate after transplantation. There was no evidence of tumor formation or any other safety issues. And in our efficacy study, we tested 4 different lots of dopamine precursor cells generated from 4 different Parkinson's patient donors, which are cells representative of our drug product. And these cells were tested in a rat hemiparkinsonian model of Parkinson's, where one hemisphere is devoid of dopamine due to a chemical lesion. And this leads to a rotational imbalance when amphetamine is administered. We demonstrated that each lot of the dopamine precursor cells resulted in significant improvement in rotational imbalance over the 28 weeks, with some rats returning to baseline. And this indicates the ability of the cells to persist, to mature into functional dopamine-producing neurons that maintain their phenotype over the 6 months of the study. And we also demonstrated feasibility of the delivery of the cells to the correct location in an NHP model. And lastly, we demonstrated we can transfer the process into GMP manufacturing. We have potency assay candidates, and we've got an approved device for delivery of the product. And data on our NHP study will be presented later this year at the Future of Parkinson's by Marina Emberg. And quickly wrap up here, tell you a little bit about our clinical plan for our ANDP-001. We've got a phase 1/2A clinical trial with 2 cohorts evaluating safety, tolerability, and dose response. And subjects from our screening study, which has already started, will be enrolled into this trial. We'll have multiple readouts for both primary and secondary objectives, some of which should give us an indication of biological effect. And so, in summary, we've got our first IND cleared with a pathway for approvable endpoints for a future pivotal trial. Our manufacturing campaign to support the clinical trial is ongoing, and we've already made product for several patients. We expect to dose patients next year with some initial clinical readouts by the end of the year, and we're executing on our vision to be the leading cell therapy company.